Hi, everyone. Welcome to the fourth day of the Biodiversity Genomics meeting, and welcome to the Early Career Scientists Forum. In this forum, our goal will be to discuss the core issues around the early career scientists at the moment, especially with the COVID-19 pandemic. And I'm Joanna. I'm going to introduce myself very briefly. So I'm a postdoc in UC Davis. I work with Harris Lewing, and I work with most in chromosome evolution. And I'm co-sharing this session with Agostinho Antunes, and now I'm going to let him introduce himself.
Hi, hello. Good afternoon, everybody. So I'm the head of the Evolutionary Genomics and Bioinformatics group at the CIMAR, the University of Porto in Portugal. And together, me and Joanna, we're gonna host this section so that we could have a better perception with a selection of talks from early career scientists and which would foster our discussion around the topic. So we would welcome all the attendees to strongly send questions and upgrade the questions so that we can, after the talks, also being able to discuss some of these issues in more detail. So in terms of the panel of speakers that we're going to have today, we're going to start with Ksenia Jurava. She's a PhD student within the IGNITE program that you may have heard yesterday in the Giga Talks. And she's at the University of Munich in Germany. Then we're gonna have
Martha Farrell. She's now a lecturer in molecular biosciences in the University of Kent. I worked with her while I was doing my PhD, so it's a pleasure to have her in our panel. And then our final speaker
is Shu Li, a PhD student from the University of Seoul in South Korea, which will go also to motivate our discussion.
And now we're gonna then listen to the videos.
Yes, so see you in a moment.
Hey, hello everyone. My name is Ksenia Jurava, and I'm going to share today a small presentation about my early career in science. Currently, I'm a student in the LMU in Munich and part of the lab of Professor Gerd Heide. My interest in biology started already in high school, but my official first step in the academia was by obtaining bachelor's in biology in 2015 from the Department of Life Science in the Hebrew University of Jerusalem. During my first degree, I was also working in the zoo as a guide and a zookeeper, and I received for part of it scholarship from the Tisch family zoological gardens in Jerusalem. As well, I was interested in research already from my first year, and I joined the lab of Professor Avi Schmida in the Department of Ecology, Evolution, and Behavior. There, I conducted my own research in interactions and behavior of plants and bees. We were going to the field, collecting bees, doing observations, and then going back to the lab to analyze them. In my third year of the degree, my interest changed a little bit, and I started to work at Dr. Uri Gat's lab in the Department of Cell and Developmental Biology. There, I helped master students and PhDs to conduct the research in regeneration in Nematostella vectensis. The picture of this animal you can see with black background on the slide. I was helping with the rearing room, carrying PCRs, and other stuff to the research. Then I went to do my own research, and in 2018, I graduated with masters in agroecology and plant health with specialization in bioinformatics and genomics from the Hebrew University as well, but from another department. There I also received excellent scholarship. In the lab of Professor Shai Morin and the postdoctoral researcher Diego Santos Garcia, we were part of the Cassava Whitefly project. This project aims to deal with one of the 100 most damaging pests worldwide. As well, it is about food security. It aims to help small farmers growing field cassava in Africa, but not only. What we were doing is to use bioinformatical tools to analyze different genes and proteins. Specifically, my project was about the P450s. It's a family of detoxification genes. We were looking for finding positively selected positions in the proteins to see if they help the pest to metabolize more secondary metabolites of plants. You can read more about the Kusawa whitefly project and what was done in this lab in the Nature paper published. So now I'm doing my PhD in the LMU University and supervised by Geert Volheide. As well, I'm part of the ITN Ignite, which you might have heard about from Geert, who presented it here. My research is about phylogenomics of non-deleterian animals, and you can see the nice illustration summarizing it by Max Telford, who is also part of the ITN. You can see here two competing hypotheses. One shows the porifera in orange on the bottom being the first to branch from the rest of the animal, and the other one in blue, the Ctenophora, being the first to branch from the rest of the animals. And if you ask yourself, why is it even important? So there are animal traits that might be different and have different evolutionary story. Depends on who was the first to branch from the rest of the animals. Neurons, muscles, guts, and other things that we would like to know. How did they develop? And for that, we need to know who was the first. So a little bit more about the ITN Ignite. It's all about being international network and having a lot of education and training events. You can see in the picture in with the flags that we are many from different places. And the first idea for this ITN came up during the GIGA conference. I have the honor to be student representative of both GIGA and Ignite. And it's given me a great opportunity to be involved in planning workshops, conferences, to talk to different researchers, PIs, and not only other people from the industry that involved in this consortium. And this is a great opportunity to have many collaborations and meet new people from the field. So I'm really happy to be part of it. And I know it will help me a lot in the future in my career. So with that, I would like to say thank you to the organizers for the invitation to present here today. Thank you to you for the attention, and I will be happy to answer questions.
Hello, I'm Dr. Martha Farrell. I'm a lecturer in genomics at the University of Kent in the UK. I started my undergrad in 2000 at the University of Barcelona in Spain, and I graduated in genetics. I spent 9 months in the University of Tuscia in Italy during that time. After my undergrad, I did my master's in cell biology at the University of Barcelona as well, and I spent 4 months in the University of Bari in Italy. When I obtained my master's in cell biology, I started my PhD in genetics at the same university. And during that time, I spent 10 months at the University of Yale in the US. After I obtained my PhD, I started my postdoc position at Aberystwyth University in the UK. And then I started my second postdoc at Royal Veterinary College in London in the UK as well. In 2018, I started my independent position as a lecturer, that is similar to an assistant professor in the US. And I'm currently a group leader in University of Kent in the UK. My group works in comparative genomics, and we are particularly interested in the role of structural variants in speciation and adaptation. Thank you.
Hi, I'm Charlie, PhD student in Seoul National University. Thank you, the committee, for giving me a chance to present my experience in Genome Consortium. In this presentation, I would like to introduce chance I received from Genome Consortium. Let me start my presentation. VGP collaboration gave me chance to find interesting result. As a trainee, I joined the VGP assembly working group, and I learned how to build high-quality reference genome assembly. After then, I wondered how many genes are improved by VGP effort. My team focused on erroneous, missing, and duplicated region in previous assembly, which are corrected in VGP assembly. My team found false gene losses and false gene gains are associated with GC content and several features, including discordant leads, respectively. Based on these results, my teams are preparing companion papers. I think it would be harder to get this chance if I didn't join Genome Consortium, like most PhD students in South Korea. In South Korea, most PhD students started their course to be professor or researchers, and most of them achieved it by working at university and research institute after getting PhD degree. However, during the PhD course, PhD students are harder to work in projects related to research topic which they are interested in, because their scholarship and experimental materials are provided by research fund of their supervisor. Moreover, the research funds for PhD students are very limited, as around $4,000 to $5,000. My supervisor's projects are a little bit different from my research topic. I was interested in comparative genomics to compare reference genome assembly. In order to know how to assemble reference genome and to start my project, I joined the Genome Consortium Vertebrate Genome Project. I will introduce benefits that I received from VGP. First of all, VGP prepared research fund for sequencing and assembly and collected samples from various countries, so I can save my time and effort to find funds and samples. VGP assembly working group provided well-organized assembly platform made by DNA Mixers, and they provide online training session based on the platform. Assembly tutor taught me how to assemble chromosome level assembly. When I investigated new biological finding with the VGP assembly for the VGP breakage paper, collaborators and great scholars helped me to develop my program to visualize chromosome evolution across vertebrate lineage. On the other hand, in Genome Consortium, there are not only opportunity, but also some risk. If you want to keep your credit for your idea and data in the consortium, it would be better to share it as soon as possible. Then you might be worried about stolen your idea. Then you do not have to worry this situation, because the consortium committee will support you based on VGP policy. Additionally, if your findings are enough to make content for on research article, you can take your PI as a companion paper. If you need more information for the VGP, then please search VGP Vertebrate Genome Project in Google. I deeply appreciate my co-supervisor and collaborating group. If you have any question, please contact to me via email. Thank you for your attention.
Thank you to our panelists for sharing with us the diversity careers that they've been having till now. We are still waiting for questions from the audience, so maybe I can ask the first one. So during your career, what is the main challenge that you feel you've faced, especially during like the first years of your career? Maybe Ksenia, you can start. Unmute yourself first, please.
Yeah, sure. So hi, everybody. In my way and beginning of things, it was difficult, I think, as for everyone, because you're not sure how things happen and what to do. But then you tune in and you get advice from people who've been there already and learn how things works and jump in and do what you need to do in your research or studies and so on. So this is briefly it, I think.
Maybe Martha, no?
Yeah, I think that at the beginning of my career as a researcher probably was that I had to retrain from a completely wet lab person that I did my master's, it was completely wet lab, to then starting my PhD on bioinformatics. So probably retrain myself as a different scientist. That was one of the main challenges that I had. And also, as Ksenia said, so training basically was the main obstacle at that point of time in Barcelona, where I was. There was not that many things going on bioinformatics, so it was difficult to begin with. But yeah, I would say that's the main thing.
Sure.
Yeah, at the beginning of my graduate school, so I'm interested in human evolution, and I would like to know the microevolution in human that was different from the other animals. So I wanted to perform the comparative genomics between human and the other non-human animals. So the training in my university, my university provides very good good coursework to learn the skills to analyze the genome, and the VGP consortium and Bird 10K consortium provide me some opportunity to analyze real data. So that was my beginning of my studies.
Mm-hmm. So we actually have now one question specifically for Chul. They ask if you can talk a little bit more how you did get involved with the VGP.
Yeah, my study is fortunately my professor involved in the Bird 10K project. So fortunately I can involve with the Bird 10K at the first phase of the Bird 10K project, and then I contact with Eric and Eric Jarvis. He is the organizer of the VGP group, and then he was being my supervisor from that time. So I'm working on the VGP from the start of the VGP project, and then yeah, now I'm still working on there. Yeah.
Well, we have here a quite interesting talk from Peter Holland, which I think you could all answer, and I think both me and Joanna, we can also add something at the end. And Peter was mentioning perhaps you could suggest one thing that senior researchers could do better to help early career scientists. So I suppose you will have different perspectives. You are also coming from different places now, so I think that this will also be more comprehensive. So perhaps we could start with Ksenia, and then following with Martha and Shu, and then both of us, me and Joanna, could do a remark as well.
Yes, so in my experience, I had very nice postdoc who guided me actually through my master thesis. So the senior researcher gave the option to one of his students to guide me, and I think sometimes it's better to do this step and let students who have more time to guide the younger students in the lab, because sometimes there are a lot of things to do apart from guiding your student. So that can be very helpful to be more people or someone who has more time leading project, because in the beginning students need a lot of help to go into details and understand what they need to do. So I think time management in this case is a key to success. So making sure that there is enough time for the students to go on and do the research and get the help they need. So this is from my side.
Yeah, I mean, as a new PI, I guess that what senior researchers could do maybe, and I'm very lucky because all my mentors that from all the different institutions that I've been, they are also on that. But probably help us on achieving, on getting the first grant. So getting the first grant as a new PI is the milestone, I would say. And getting feedback on the grant applications, for example, that's something that I find really helpful. So probably even senior researchers from different institutions working in similar fields or slightly different fields might be interesting to create some kind of help available for that. So get feedback of grant writing, that would be one of the key things that I would suggest as a new PI.
Sure, could you?
Yeah. My PI is in Seoul National University. He, as I mentioned in my talk, he has a different topic, and even if he don't has even if his main interesting is domestication, but he provided me to get a fund for the VGP consortium, and then I can sequence the Korean indigenous species. So yeah, and I think the supervisor can provide some good opportunity for his student. So yeah.
Very well. Joanna, what you would like to add here?
Well, I agree with all of them. I think during my PhD, it was important to have like a good support, even for my supervisor, and in my case from Martha too, helping me that way in the day-to-day basis. But it's also, I think it's important to have like a good network of students that are in the same situation as you, that can give you advice how they solve the problems that they face every day. And now kind of finishing the postdoc path and thinking about going to an independent career is also important to have the chance to write your own grants and get used to that dynamic as well.
Very well. Well, I'm just going to add one line here. I think from my perspective, education and training, it's really important for the young researcher. So I think with that background, he would be able to do well, even if he stay in the same place or if he go abroad and would have different challenges. Basically, if you are well prepared, it's most likely that you do well in the future. And that certainly will help you if you are eager, motivated, hardworking in getting the chance not only to having a nice collection of papers, but also, and that will be really relevant for establishing an early career, trying to get grants and funding to support your ideas, to start engage with the possibility of doing the things from your home. And I think this is certainly something that I would place as really important. But as we will discuss here, there is other things now. And hopefully together we can place some comments here that could allow us all, either from students or more advanced researchers, to think clearly in the issues of the early career scientists, which is particularly important now in this COVID-19 pandemic situation. Very well. We have here, Joanna, maybe you could upgrade here some of the questions. And
yeah, So I'm going to ask this one to Martha because I'm also very interested in this. So how did you know you were ready to transition from a postdoc to setting your own lab?
Oh, that's a really good question. So I had for some years, so I was a postdoc for 6 years, 6 years, always with the same PI, but in different institutions. And the last couple of years of the postdoc, I had already developed my own ideas, and I wanted to explore these ideas. And I started applying for independent fellowships, and he was extremely supportive. So I had down to Dennis Larkin on this. And I was not successful on the fellowships, but I wanted to develop this further. So I knew that I had my own ideas, and I wanted to carry with them, and I started applying for lecturer positions. So as I said, lecturer positions are equivalent as an assistant professor. So it's a tenure track degree. Yeah, tenure track. And yeah, I mean, that's why probably when I knew that I wanted to start my independent position, because I had something specific that I wanted to ask, and that it was different from what was going on in the lab and the research interest of the PI. And I was already supervising some people in the lab. So I had some experience on supervising undergrads mostly, and some master students. And it was going okay. So I thought that I would be good enough to get a PI position. And yeah, I think that's probably when I knew that I was ready.
Very well. So I cannot resist. There is an interesting question that was anonymous placed here that says, how can you do work and life balance, specifically now that you need to publish or perish? I think this is a quite interesting questions now, and that was some of the topic also that I previously discussed with Joanne, you know, the opportunities, also the equally opportunities regarding gender. And there is difference, obviously, and we try to accommodate here with different perspectives, and the male or female perspective is slightly different. And getting a family, and for ladies having children is particularly demanding. For men as well. No, I have 3 daughters, so it's a huge challenging, but for a lady it's particularly hard in that sense. And I would like to have your perspective. No. In one side, this huge need to publish and to be successful and to collect money and to have grants, and at the same time to have your balance in life, your family, or establishing your family. And this is particularly challenging for the early careers researchers that have this dilemma, you know, how to sacrifice my life or to have a balance between my professional life and my real life. No? Because after all, and now I think with this pandemic perspective, we know now more than ever that family and balance, it's a lot. So perhaps we could start maybe with Ksenia or Martha to have your more sensitive perspective on this question.
Yeah, I mean, since the pandemic, obviously I've been working from home, so the university was shut down. And I could probably say that my work-life balance, I'm not a good example of how it should be done. Yeah, basically this. So because it's a bioinformatics group, and my group is relatively small, we can work a lot from home, and we've been doing that, which I don't think it's a good thing to do. What I try to do for myself is always try to find some hours on the day, or some plug down some weekends to not work. And what I'm being very conscious is how I deal with my students, because I don't want to set myself as an example. So I try always to not email them outside of office hours, let's say, or email them on the weekend. I might write the email, but I set some kind of timing so that they receive the email at a normal time. We work using Slack, but I told them to stop Slack during non-working hours, although I keep answering and asking questions. So Yeah, I think that now with the pandemic, it's even worse, the work-life balance. And as a family as well, so it's a complication that it's there. I don't have kids, but I think that it will be really difficult. I have colleagues that they have kids, and it's becoming just unbearable right now. Also, one of the things that it's not very commonly mentioned is the two-body issue that scientists we have. So the thing that when I married, when it was very difficult for me and my husband to find a position that was relatively close to each other. And this is not a thing that is most taken into account in job searches or even from panels perspective, that this is also a thing that might imbalance your work-life balance just because your husband or your partner is somewhere else. So your mental health might be a little bit not ideal in these situations. But I think as an UPI, I'm trying my best to not be myself an example. And because I don't want my students to have a bad example, I'm forcing myself to have a better work balance that I used to have when I was a postdoc. So I think that for me, just trying to be an example and forcing myself to have this work-life balance is working better. But COVID-19 probably was is setting this a little bit back. But yeah, I'm trying to go back to this.
Yeah, I would like to add to what Martha said about finding positions and everything, that as a student from the student side, I moved to Munich from Israel and kind of left the center of my life for 3 years for the PhD. And it's difficult to maintain a balance when you know that your friends and your family all behind and you're now kind of isolated, not because of COVID, but of the studies. But I think the first good thing is to realize that you're not alone, and when you talk about it with people, it kind of put you back to think about, oh, okay, so people need to spend time on something else, maybe I do as well, I need to find a hobby or something, and then to try to keep it. So it's kind of two works. One work is to be good at your research and to keep and go forward, but the other work is to stay with what you like and do hobbies and have your time for things you really care and want to do. So it's two parallel works to keep go on both directions and try to maintain it. Not easy, but we need to do it.
I also want to add something. So in my case, my family are very supportive for my graduate school. So so I can work in VGP, awesome VGP group. So it is based on, so yeah, it is really, my family give us a very good chance based on their patient. So yeah, and I also very had to find some balance between my course, my study and my life, family life. So, but I always want to set the top priority for my family, but I think it is always I ask to my family to help, and they accepted my my request. So yeah, so it is not easy to meet the balance between study and family life. So, I please give me a good yeah suggestion. Yeah, to make us some balance.
No, balance is all in life, no, and we need to keep it for a long time now if we want to be in research, and research is often a very exhausting field. So it's certainly good to keep the balance. So Joanna, perhaps you could go ahead with some other.
Yeah, so there's interesting one, since we talked about having a network of people that are in the same situation as us. Mark says that he's very excited for the discussion, and with COVID-19 pandemic, if we think it could be interesting to create the early career scientists meetings so we can know each other we can meet each other, at least virtually.
Well, I think this is clearly a very interesting idea. And I think this meeting as well is giving us a new perspective how we can do it remotely, but still alive. And I think it's certainly something that we need to redefine. In my opinions, these virtual meetings can be a major advantage for early career scientists, because many often they may not have the funding opportunities to go and to attend certain meetings. And obviously, by doing remotely, this makes this information available to a wider audience, which is not restricted by funding. And so I think it's really really important for the young researchers, and hopefully a lot of the attendants in this meeting are young researchers. But I would like to see what suggestions some of you may have for these possibilities of early career scientist meetings. I don't know, maybe Martha or Cassini or Shu, maybe you want to add something in this regard.
Sure, I can say that, as I mentioned, there is the ITN Ignite group, and this is a training event for early researchers. And we do try to have meetings, virtual meetings, to keep this group now, because we had several workshops, but now it's not possible to meet in person. So we try to do it virtually. And it's a good idea maybe to divide early researchers into other groups of interest, because maybe if we meet all together, it's too much people at once. So there is a channel in the Slack for this forum, and maybe we can do subgroups and we can continue and meet and discuss topics or present our works. And if PIs can join, that will be great. So we can have some perspective from them on our research or different topics that people are interested in. So it's definitely a good idea, and we should try to do something with it.
Well, I think in this case, I would just add one sentence here to the work of Cassini. You know, she's involved in a ETN PhD program, and this is certainly a major advantage relatively to other PhD programs, which may not have this sense of group and connection. Even now, we need to do it more remotely, but even so, I think it also provides balance in terms of research. And I think this is quite a nice point.
Yeah, I mean, I think that this PhD group sounds really amazing, and I would suggest something probably similar for new PIs as well. And my experience is every time that I meet new PIs, we are facing all the same problems and the same uncertainties, and I think that grouping all together, it's always nice, and then we can learn from each other. But one of the questions that it was before, how senior researchers, more established researchers could help, I think that maybe being involved in these new PIs meetings can also be interesting, because what I find being more complicated right now is not the research itself, it's probably managing people and having experienced people that they managed a lot of PhD students or postdocs. That also will be an advantage for new PIs as well, how they encountered or how they resolved some of the issues that they had on setting up a lab, and how also to navigate the politics on behind grant funding or consortium as well. So having more experience on that, it's always welcome. So probably setting up this kind of activities by experience level will be really interesting.
I'm going to pick here a question that was placed by Joe Lopez, which I think is really nice and was upgraded by several people in the audience. And Joe mentioned what do you think that early career networks could be especially important, well, for all scientific disciplines, but in particular for genomics? And I think here you can have a lot to add. No, clearly, genomics can be overwhelming, and in this genomic era, doing the work just by yourselves or just a few people can be challenging. And in particular, in these large genome projects, we need to congregate efforts. And so the networking is particularly relevant. And I think you all have experience in this sense, although probably slightly different, but it will be important to know more about it. No, we could start now with you, that has been involved also in these networkings previously, and since he didn't comment the last question, I think we could start with him.
Yeah, for the networking and collaboration, so I think genomics, I think it is the very important point is the data sharing in the group, the genomics group, including VGP. So VGP widely open to give us some open opportunity to access the data. So in the genome arc, the genome arc of the VGP is we uploaded all of the raw data and assembly, so we can access the data, and everyone can analyze the data, and everyone can share their data in our group. So I think the senior group have to make some field to some place to share their data, and have to make some community to discuss their ongoing studies. Yes, ongoing studies of early career scientists. So I think we need some we need help from many senior scientists. Yeah.
Any other suggestions from Cassini or Martha?
Yeah, so I can say about the Ignite training network, which came out during a Giga conference as idea and then become reality. That's the great advantage of such thing is that I feel like I have a lot of people to ask questions. So I came to this PhD as a newbie to genome assemblies, and I did transcriptomics, which is a smaller scale, of course. And here I have people who know different methods and tools, and it's overlapping a bit with what Drew said, that we need senior researchers to guide us. So here in this network, it's all about training and having a lot of people together focusing on genomics as well. And it's very helpful to know that you have someone to ask a question about what you need, and you have someone with experience. So it's not about only databases and protocols, but it's actually to talk to people and get answers to your particular case. So the network is great, and thank you for this network because it helps. And also there are resources out there that people can read, and we try to establish a channel and keep all the materials from our workshops, so other students can go there and learn from the protocols we have for assemblies and different methods for genomics.
Very well. So Joanna, we still have a bunch of questions. So perhaps we can take another one and try to see if we.
Yeah, so I'm gonna pick this topic for discussion that Marcela Uliano posted. She said, science is done by people. A good group leader will put the needs of the people in her or his group first Any thoughts on that? I think that's the way it should be, right? And I've been lucky enough that my bosses have done that.
I completely agree with his statement. And as a new PI, I mean, it's the first thing that I thought about my group, how I wanted my group to be and how I wanted to behave also with my students. And it's no longer my research career that is in place. It's a career of the PhD students and the postdocs that I have in my lab. So it's trying to coordinate everything and advance all of us together, working as a team, not only as a selfish person. I don't think that's how it should be done. I think that as I said, I've been very lucky in all the previous labs that I worked at. All of them were exactly the same philosophy. And I think that this needs to be broad spread to everybody. So I think it's how ideally it will work everywhere.
Any comments from our PhD student?
So I'm lucky to have a very supportive PI, and usually when I'm coming with ideas, he's like, go ahead and do it. So I think it's important because I feel like I have place in his lab. So even if it's his lab, I still have my own place and I can do what I want to do and have my ideas developed in new directions. And it's great. It should be that way, I think.
Anything to add, Shul?
Yeah, I think I'm very I'm lucky guy, so because I met a very good co-supervisor. My Korean supervisor, Professor Hibar Kim, provided me freedom to take a different topic in his lab as a PhD student. So I think that is very good opportunity for me. And the other co-supervisor, Professor Eric Davis, he also gave me a big chance to work with a very good collaborator, including Joanna and I would so, and yeah, in the other VGP members. So I think that meeting people and in the meeting people is we can find some good chance. So I would like to promote the collaboration for early scientists, early career scientists like me. Yeah, PhD students.
Very well. Well, I will add some comment here as well. I think science is a competitive field, but if it is too much competitive, it lacks fun. You know? I think most of all, and if we want to do science for a career, for life, we need to have good relationships. And for instance, in my perspective, I've been working for over 20 years with colleagues, for instance, from the States when I stayed there a long time ago, and still working in a current basis with them. And at the same time, in my own group, I have researchers and that have been staying for over 10 years now. And even when funding is not easy, and we need to be skilled in getting the opportunities for the young researchers so that they can stay, because while going abroad is really nice and welcome, but not everybody is able to do that for personal issues. Now, if they have family, it's not always easy to go abroad. Some are able to do it, but some others need to find the opportunities to be able to persist, even though trying to do good science. And I think that's possible too. And I think this is a question for many, I'm sure, of the audience. So I think it's possible, and people can still do things differently and in very good ways. So most important is a good relationship. And I think that's the basis also to do long and persistent good work. So I would try to pick another question here. So basically there was a question here mentioning relatively to the importance of playing around with both wet lab and more dry lab. No, in these cases, in this pandemic situation, clearly this could be an option. From my side, the group I coordinate, we have both wet lab and dry in silico options, but it's not always the same for everybody. So I think, and among all of you, I think there is different experience. Some of you will be more engaged with bioinformatic work, some also with a good amount of lab work. And I would like to have your perception also, because in reality, in terms of balance for science, both things are important now. So we need sometimes to play around with both. But now, and I think for the early career scientists, this is something that also would be important to discuss a little bit.
Yeah, for me, for the Korean indigenous species project, I get a fund for the PGP, but there is no professionality to identify species, to perform species identification and to collect the sample from the sample from isolated tissue sample from the individual. So I really I need the collaboration from wet people. So in the field, they work in field for morphological analysis and to know their habit to know their yeah. So I need the collaboration initiate my project. So I think the important thing is the collaboration for the dry lab people.
Well, I don't know if Martha, and we are running out of time, but maybe quickly, Martha or Ksenia.
Yeah, I mean, I agree with Jules. So trying to have a collaboration with a completely wet lab, it's always useful. In my case, I'm trying to run a mix. Some people are wet lab and some people are bioinformatics, but then we still need field scientists as well. So if we want to get samples, we will need field scientists, and I'm not an expert, and I'm not wanting to be an expert on going to the field and collecting samples. So that's why I think that we need to rely on people that have a lot of experience on these fields. And yeah, I completely agree. It needs to be a collaboration between different types of labs. And now in the COVID-19, I had to move all my research to bioinformatics, obviously. And this impacted at least 2 of my students. So that they were wet lab and they had to be moved to bioinformatics a little bit reluctantly, but yeah, they enjoyed it at the end.
Well, Joanna, I don't know if you want to pick any other question quickly, or we need also to make a balance of the discussion, although it was so many.
I think, yeah, so maybe we can just summarize a little bit of the discussion. For me, the main thing was that you should then create the networks for early career scientists at different levels, right? So maybe you can keep the Slack channel that we have for this forum from the meeting and try and develop after that. And I see that we have a lot of questions still here, so maybe we can move the discussion there. So if anyone else wants any advice, I see a lot of them for Martha's firstly, trying to figure out the balance and how to look for jobs and lecture positions. So maybe people can ask the questions there too.
Well, there is lots to be discussed in this subject. I'm just browsing the questions, and there is a lot of them that are really interesting. I would love to go along them, but the time here it's a relatively short. So I would like to thank the speakers for this opportunity to see better your work and present your work to the audience. A big thanks for the audience that have been really enthusiastic. They placed a lot of questions. I'm thrilled to see how interest this topic is for all of you. And it seems, although we don't know exactly the audience, it seems to be there is a lot of young researchers here, which as some others mentioned here, Aris mentioned that's the future now, and we need all of you to change. And as some of the questions were placed, to make these stories a success and not just a question of lucky. And I think it's among all of us to change this path. If we can communicate better, if you can share our experience and trying to do things in a way that we could do a good work without being, I think, over competitive, which sometimes can kill these human relationships. And science needs to be done with quality, but also with balance. And most of all, it's done by people, so it needs to be really pleasant. I think that's my opinion. And I think biodiversity genomics is in that target. So I don't know if Joanna or some of the speakers would like to add anything else. We are running out of time. And
yeah, so I just want to thank you all, our panelists and also the attendees. And now we can move to the plenary talk from Carolyn Hogg, so we can have another interesting talk.
Very well. Thank you, everybody. And feel free to contact us and use also the Slack channel to place more questions or know more about some of these topics that we discussed here. Thank you very much, and it was really nice to have you here.